In this one, we are going to take a look on signaling molecules. So signaling molecules is a topic given in our genetics course. And once we are done with this topic, signaling molecules in humans, then we will be ready to study the topic defects in signaling pathways and their consequences. So let us start with the signaling molecules first. So to begin with, what are signaling molecules? So signaling molecules are those low molecular weight molecules which play a role in cell-to-cell communication. So one of the cells will be releasing this substance, the other cell will be responding to this substance, but between the release and the response, there are a lot of steps. First and foremost, this released signaling substance will travel to the target cell. Second, on the target cell, it will bind to a receptor. Upon binding to the receptor, it will initiate a process. And at the end of that process, there will be a response from the cell which received the signal. And the cell that responds to the signal is known as the target cell, and the cell that released the signaling substance is known as the signaling cell. The target cell always has got a receptor, and the signaling cell releases the signaling agent or signaling substance. So that is the basic relation. If I draw a small diagram here, so what you see here, that let's say that there is a cell that is releasing the signaling substance. So this is a cell, and this cell is releasing the signaling substance, and this signaling substance will get transported. And how does it get transported will totally depend upon what is the nature of this signaling substance, but eventually it will get transported to the target cell. So now this other cell is a target cell, and the target cell is a target cell because it has two things. Number one, it has a receptor. So this is the receptor that I'm drawing. So it has a receptor to which the signaling substance will bind. And upon binding to the receptor, within the target cell, there will be a specific process that will be triggered. But at the end of this specific process, there will always be a response. So basically, if you now look at it, so the steps that you are looking at, these steps are essentially, number one, release of the signaling agent. Or signaling substance. We are using the term signaling agent. Second part is transport. So how far it is transported within the body? Sometimes it has to travel really far. In case of endocrine hormones, the substances which are signal, that is the hormones in this case, they travel through the bloodstream. And from the gland to the target organ, they have to cover quite a distance. But in some cases, the signaling substance will be released by a nearby cell. That is the case of paracrine signal. Sometimes the signaling substance is going to be released by a cell which is just the next door cell. That is the case in case of neurotransmitters. So it entirely depends which signaling agent we are talking about. Based on that, the distance and the mode of transport question will be coming. This is the third part that you are looking at. The third part is where the signaling agent binds to the receptor. So this third part is receptor binding. All right. And this fourth part, that is the internal process that is leading to the response, is known as signal transduction. So signal transduction is a term that you can use for all the processes that take place between the binding of the signal to its receptor and the response coming from the receptor cell. So all the in-between things that happen from this point, that is receptor binding, to the response. So this fourth step is signal transduction, and fifth part is the response. All right. So that is how. And this cell is the target cell. And this cell is the signaling cell. So the substance that is released by the signaling cell is called the signaling agent or signaling substance. Okay. And in this chapter, we are going to see what are the types of signaling substances, and out of these types, what are the major functional categories that we are looking at. So I hope you have understood this basic concept of what is a signaling substance. Okay. So if you look on the screen now, so a couple of important things that I would like to highlight is an essential component of complex system of cell-to-cell communication that regulates various physiological processes. Signaling molecules are responsible for communicating messages between the cells in the body, and that ways they help in maintaining homeostasis and coordinating the functioning of various organs. Now we are coming to its typification. So first typification is based on the location of action, that where is the signaling agent meeting the receptor. So it depends upon what is the chemical nature of the signaling agent. So think of a peptide that is large in molecular weight, that is strongly polar, that it has some internal charge asymmetry. In some cases, it may have a net charge also. So in all such cases, the signaling substance will be finding it very hard to cross the plasma membrane. So for such signaling substances, it is important that the receptor is located within the plasma membrane itself. And the receptor's signal binding domain is towards the exoplasm of the cell. Exoplasm is what is outside of the cell. So when the signaling agent binds to such a receptor and initiates the action, so such a receptor is going to be called cell surface receptor, because these receptors are located within the plasma membrane, and plasma membrane makes the cell surface. So for peptide hormones, amine derivatives, and most of the hydrophilic substances which have large molecular weight, the receptor is located on the cell surface. So that is a very big category of signaling molecules which act via the cell surface receptor. The second type of signaling molecule is those signaling molecules which are hydrophobic, which are lipophilic, means lipid में उनकी solubility ज़्यादा है, water में उनकी solubility कम है, and besides they are low molecular weight. So for example, steroid hormones, thyroid hormones, not only that, the low molecular weight substances like nitric oxide and so on. So these substances, the nitrous oxide, not nitric oxide. So these substances are the substances which can easily cross the plasma membrane. Therefore, it makes sense for their receptor to be in the cytoplasm. In some cases, the receptor can even be present within the nucleoplasm. So for thyroid hormones, the receptor is present in the nucleoplasm. For steroid hormone and also for nitrous oxide, the receptor is present in the cytosol. Okay, so here we have, first and foremost, we have got extracellular signaling molecules. They act outside of the cell and they have cell surface receptors, and they require multi-step transduction pathways for the cellular response. So examples include peptide hormones and growth factors. Growth factors क्या करते हैं? They stimulate the cell to divide. So they are classic cell surface receptor acting signaling agents. So these molecules bind to a specific receptors on the cell surface. They trigger a cascade of events inside of the cell that ultimately leads to cellular response. Take a look here. So now you can easily see this. So this is the peptide hormone. It could have been a growth factor. It could have been epinephrine, and all of these, they act on a cell surface receptor. This is the receptor. The receptor is always a protein. The signaling agent may be a protein, may not be a protein, like in steroid hormone, in thyroid hormone, it is definitely not a protein. But even then, the receptor is going to be a protein. So this binds to the receptor, which is a protein, and notice that receptor is present on the plasma membrane, correct? So now that the receptor is present on the plasma membrane and the cellular response is going to be elicited by some molecule which is deep inside the cell. So in between them, you need a number of proteins to create a series of communication. And this series of communication is going to be called the transduction pathway. And notice here, so there is this protein, then this protein, then this protein, and they keep on activating each other. And eventually the final protein will go to the site of action where the cellular response is to be elicited. In this case, there is a gene whose expression is to be activated. So this substance goes into the nucleus, will bind to the gene, and will stimulate the gene expression. In this case, gene expression was the desired cellular response, and that cellular response happened. So this is the classic way in which the extracellularly acting signaling molecules will be working. Now we are going to talk about the signaling molecules which work intracellularly. So the signaling molecules which work intracellularly, they have got four features. Number one, they are water insoluble. They are lipophilic and lipid soluble. They have low molecular weight, and they are non-polar and uncharged. By the virtue of all these four things combined, they can cross the plasma membrane easily. Since they can cross the plasma membrane, they will be meeting their receptor either in the cytoplasm or in the nucleoplasm. Cytoplasmic receptor applies to the steroid hormones. Nucleoplasmic receptors apply to the thyroid hormones. So they act inside of a cell, typically lipophilic or low molecular weight non-polar compounds, such as thyroid hormones, steroid hormones, and nitric oxide. These molecules have receptors in the cytoplasm or in the nucleoplasm. So most of your competitors are not going to mention the nucleoplasmic angle. But when you study the role of thyroid hormones in amphibian metamorphosis, the molecular mechanism, you can recall that the receptors were present in the nucleus. So the receptor can be present in cytoplasm or nucleoplasm. And when the signaling substance and receptor, they form a complex, this complex is mostly the transcription activating complex. So moving next, so serves to activate transcription, and that leads to changes in gene expression. Take a look here. So this one shows the working of steroid hormones. So take a look, the steroid hormone, that by the virtue of being low molecular weight and hydrophobic, lipophilic, unpolar, could directly enter the cytoplasm. In the cytoplasm, the receptor was present, and together the hormone receptor complex gets formed. Then this complex goes into the nucleus, and then it will be binding a target gene. So in the earlier case, you noticed the first case, the hormone receptor complex remains on the plasma membrane, but that activates a set of protein. And in this case, hormone receptor complex itself goes into the nucleus binds to the genetic material and activates the transcription. So these are the two major categories of signaling agent based on the site of action. Now we are going to get familiar with four functional categories of signaling molecule, and that will complete this chapter. So four functional categories of signaling molecules happen to be hormones, neurotransmitters, cytokines, and growth factors. Let us learn about them briefly. So, hormones are the secretable substances which are released from specific organs known as glands. And then the hormone will be traveling through the bloodstream to reach the target organ. And then it will be impacting the functioning of the target organ. So, hormones are chemical messengers released by the glands, travel by the bloodstream, and govern the functioning of a specific body organs. So, that's what hormones are. Hormones are very vital in maintaining the metabolic homeostasis. So, chemical messengers secreted by endocrine glands into where? Into the bloodstream and act on cells or overall organ to regulate various body function. And the body function can be metabolism, growth, development, and reproductive processes. Hormones fall into two classes. Class 1 is lipophilic hormone, steroid, thyroid hormones, and class 2, which are peptide hormones. So, hormones can be classified on their chemical structure, such as steroid hormones, peptide hormones, amine hormones, etc. Okay, rest is example. But in Harper's biochemistry, you will get to learn this, that there are two classes of hormones. Okay, and the two classes of hormones happen to be class 1, that is lipophilic, and class 2, हाइड्रोफिलिक. अगर इनके बीच कुछ हमें डिफरेंस करनी हो तो व्हाट आर द डिफरेंसेस दैट यू विल बी एबल टू डू. सो ऑन class 1 they will need plasma protein in the blood for transport, and class 2 is going to need no plasma protein for transport. Right? So that is the first one. Because the thing is that class 1 is lipophilic. So any lipophilic substance, if it has to travel in the bloodstream, that is largely water, needs something to hold it and move through this aqueous medium. And this is hydrophilic. So this does not need any plasma protein. Where is the receptor? Here the receptor is intracellular. Here the receptor is cell surface receptor. Here you have got short transduction. You remember that here the hormone receptor complex directly gets into the nucleus to activate transcription. Here it is multi-step transduction. So these are some of the crucial differences. Then you can always bring in examples, right? So that is about hormones. Now, neurotransmitters are the chemical substances that act on the adjacent neurons. They help in transmission of electric impulse and they are basically the signaling agents which will be working upon the ion channel linked receptors. Ion channel linked receptor का मतलब क्या होता है? These are the receptors which are connected with an ion channel. When the receptor is bound by a neurotransmitter, the ion channel will be activated. And as the ion channel is activated, the neuroelectric impulse will be transmitted. So these are chemical messengers which are released from neurons and act on other neurons or target cells to convey signals and regulate the activity of the nervous system. So here you have dopamine, serotonin, acetylcholine and all. And like all other signaling agent, they act by binding to the receptor on the target cell, which triggers a cascade of electrical signals in the form of action potential. And as a result, there is an ion channel that will be activated. So here we have got the drawing here. So notice here that this is the synaptic junction, one neuron and the previous neuron. So when the action potential reaches the presynaptic terminal, so there will be depolarization and there will be calcium ion that will be entering. And the beauty of calcium ion is that it will be causing the fusion of the secretory vesicles with the plasma membrane. So these are the secretory vesicles which are already there in these cells and they are packed with neurotransmitters. So they will come and fuse with the plasma membrane. As a result, the neurotransmitters will be released in the intercellular space, which is rather narrow. And pretty soon they will be binding to these ion channel linked receptors. So the neurotransmitter receptors are mostly ion channel linked receptors. And once these receptors are bound by the neurotransmitter, you can see the ion channels are activated. And as the ion channel is activated, it will be causing the change in postsynaptic membrane potential. And that is how the action potential gets propagated to the next cell and to the next cell. So this is how neurotransmitters work. Next is cytokines. So cytokines are signaling agents within the immune system. So they are released by the cells of immune system and they act on other immune cells to regulate the activity of the immune system. So during the COVID pandemic, one of the big factor that led to the complication was cytokine storm. So in this case, the patient's body immune cells will be releasing a terribly high amount of cytokine. And that will be activating the immune cells like anything. And these immune cells will then lose the distinction between self and non-self, and they will start attacking the self cells. In this process, there will be clotting, there will be damage to body's own tissue, especially the lung, and this will rapidly deteriorate the condition of the patient, often leading to death. So cytokine in limited quantity is very much needed because cytokine in that limited quantity will be balancing the activity of different immune cells by activating them optimally. But this was one pathogenic condition where excessive cytokine threw the immune system regulation totally out of the window. But of course, that was a very unique kind of pathological condition, right? So examples of cytokines include interleukins, tumor necrosis factor, interferon, and they work by what? By binding to the receptors on the target cell, and then the target cell will trigger a cascade, and that will be activating the target cell, lead to a response in the target cell. And finally, you have got growth factors, which are proteins secreted by cells of the body and act on target cells to promote cell growth, differentiation, and survival. So generally, growth factors either cause cell division or they will be causing cell survival and to some extent cell differentiation. If the growth factors are withdrawn, a lot of cells will eventually die. So that is growth factor, and growth factors get their name by the target cells which they work upon, like epidermal growth factor or platelet derived growth factor, so on. So this is what the signaling molecule functional types are within the human body. So they play a crucial role in regulating various physiological processes in the body and involved in pathology of various diseases. So that is all about signaling molecules in human body. And now logically, the next chapter is going to be on defects in signaling pathways and their consequences for human beings. So that is all in this topic.